Assalamualaikum and hi. We are from Group 4 going to present about volumetric analysis, which is titration in food industry. Before we start with our presentation, let me start introduce our group members. In our group, we have 4 members, including me, Siti Shamim Shahira.
Hi, my name is Ashley Tommy Chow.
Hi, my name is Alanisa binti Barisham.
And my name is Nuruna Suha binti Jamalul Arifin.
The topic that we will present contains what is vitamin C, introduction, basic components, theory, methodology, and conclusion. First of all, let me explain what is vitamin C. Vitamin C is a water-soluble vitamin. It is needed for normal growth and development. Also, vitamin C is needed for the growth and repair of tissues in all parts of body. The function of vitamin C, it can help strengthen our immune system, lower blood pressure, improving iron absorption, and reducing heart disease. Contribution of titration in industry are food processing, pharmaceutical, and cosmetic.
Titration is a chemical analysis procedure that determines the amount of a sample's ingredient by adding a precisely known amount of another substance to the measured sample, with which the desired constituent reacts in specific known proportion. There are 4 types of titration. The first one is acid-base, second one complexometric, and the third one precipitation, and the last one is redox.
Next, 3 basic components that are needed in titration are burette, burette stand, and titration flask.
Next, I will explain about purpose of titration. The basic purpose of titration is to determine the unknown concentration of analyte in a sample using an analytical technique. Also, titration is used to determine the equivalence point at which chemically equivalent amounts of reactants are mixed.
There are 2 types of titration that we learn, which is back titration and direct titration. So now it's my turn to explain about the differences between back titration and direct titration. Back titration is a method used to determine the concentration of an unknown using excess amount of a compound with a known concentration, while for direct titration, it's the basic method that involves the reaction between the unknown compound and the compound with a known concentration. Other than that, in back titration, 2 chemical reactions will occur, also done between 2 known compounds. For direct titration, only 1 chemical reaction will occur and then between a known compound and unknown compound. For the methodology, we choose to discuss about the determination of vitamin C concentration by titration. So, first of all, equipment needed in this experiment is burette and stain, 100 ml or 200 ml volumetric glass, 20 ml pipette, 10 ml and 100 ml measuring cylinder, and also 250 ml of conical flask.
Sample preparation. For sample preparation, we will use fresh orange juice as our sample. First, the juice is strained through a cheesecloth to remove any seeds and pulp which may block the pipettes. The setup of experiment. The burette and stand is prepared and adjusted properly. Then, pour between 150 until 100 ml of iodine solution, but 100 ml of iodine solution is preferred. You pour it into a 100 ml beaker and then you transfer it later to the burette. And step 3, remember to rinse a little bit of iodine solution into the burette before pouring everything into the burette. The titration method. Step 1. Pipette a 20 ml of fresh orange juice sample into a 250 ml conical flask and add 150 ml of distilled water and 1 ml or 6 to 12 drops of starch indicator solution. Now, titrate the sample, and the end point of the titration is when a dark blue-black color is produced because of the starch iodine complex. Step 4. The steps are repeated until the average of iodine used is found and calculations are made. Now, let's talk about the results of this experiment. The results will show that before the endpoint, the color of the orange juice was bright orange. However, once all the absorbing acid has been oxidized, a slight excess of iodine will complex with the starch indicator and the color of the solution will turn green, and this is called the endpoint. Yet, if the iodine solution is added more to the solution, then the green color of the solution will appear more darker than the endpoint result.
Throughout this topic, we already explained about the methodology, the differences back titration and the direct titration, the methodology, the introduction and three basic components. We also can conclude that titration is a laboratory technique that used to determine the concentration of substance in a solution. Titration also is widely used in analytical technique in the food industry to determine the various of chemical. Then the main purpose of titration is to determine the unknown concentration of an analyte and using an analytical technique. That's all. Thank you.